Good morning. Welcome to the Focused Ultrasound workshop on glioblastoma and the session on burning questions. If we can, bring up the slides for a second. Next slide. This is, I pulled this data off PubMed. It's in reflection of the number of publications on PubMed with the term focused ultrasound. As you can see, a steady increase. The little dip at the end is because I included this year's, but you can see this year's only partway over. We're already at a point where we are going to likely eclipse the number of publications in the preceding year. As we see this exponential growth in publications, we know that this is a emerging technology that is going to have significant ramifications across the board, not just in glioblastoma, but so many other intracranial applications. Next slide. This is specific to the number of publications where we have looked at specifically brain tumors and then glioblastoma. Brain tumors is the blue column, and the yellow column is the number of publications by year for focused ultrasound and glioblastoma. Not quite the same increase in glioblastoma as with brain tumors, but clearly exponential growth of publications over the last decade or so in this field. Next slide. So we did a study of some of the members of this workshop, and Dr. LeBlanc was kind enough, Susie, was kind enough to put together a survey, just two simple questions. The first was, what is the greatest unmet need for glioblastoma patients? And we kept the responses pretty categorical as shown here: frontline therapy, residual tumor, recurrent tumor, radionecrosis, all of the above or other. Next slide, please. This was some of the breakdown of those results. And the taller the bar, of course, the more respondents who selected that. Recurrent tumor was the category that received the most votes in terms of unmet needs, other than all of the above, suggesting that we've got, not surprisingly, an awful lot of work to do in terms of bending the curve of this disease state. Next slide. The second question was really meant to try to suggest where the next clinical trial of focused ultrasound for brain tumors and specifically glioblastoma should occur. Again, similar sorts of categorical responses that could be selected, or the last one being other. Next slide. And there was even less consensus amongst these experts that we have, and I'd love to, we'll get into this. We have some who thought that recurrent tumor should be the target, some who thought that residual tumor should be the target, some who thought frontline therapy should be the target, and others who said immunotherapy with frontline treatment. It's clear that even amongst the experts, and all of these individuals who have joined me today are truly experts in the field, that there is disagreement about what path to proceed. So next slide. So on some days in neurosurgery, I feel like this, looking out into the distance, and I see everything looks clear. It's the path is a certain one. And then as I looked at those survey results, next slide, it seems to me that the road may be a little bit more complicated. We may be hitting a fork in the road, and what now? So to answer that question, next slide, I have this distinguished group of 6 panelists. My hope is that they will shed some light on what next and where the unmet needs are. So with that, we can drop the slides, and let's, I think all of the members have been involved so far in the workshop. I won't spend precious time introducing everyone. Dr. Berger, let's start with you and your thoughts on the survey results and where you sort of see the unmet needs from a neurosurgical standpoint for glioblastoma, and where you think that the next clinical trial for focused ultrasound should begin with glioblastoma patients.
Well, Jason, thanks very much. Yeah.
And that may presume that you think that we're at a point where we should plunge into a clinical trial, although there are some that already exist. But if we were to go to a large clinical trial, where one might pursue something like that. So your thought?
Well, I think when you look at the potential opportunities for using this really new, exciting technology, I think you got to go back to the basics of what got us to this point now, meaning why are we, for me, 37 years into my career, Henry and I are the senior dogs on this panel, but for many, many years, we've just seen slow incremental progress with what we're doing. And I think a lot of it is because we just still can't define the extent of the disease. And we don't know exactly how to predict, although we're getting better with molecular signatures about how to predict the patterns of recurrence. So, you can make an argument that for each of these points in time, initial, residual, recurrent, that the technology is indicated if it can help us with the therapeutic modalities that we have at that time point in the disease. But again, I think the problem is the extent of the disease. So for example, if we could use this technology to help us deliver therapies, whether it's immunotherapy that Michael's working on, or whether it's some sort of targeted therapy that our neuro-oncology colleagues are working on, it really doesn't matter. But I think the main goal here is to try to do something we've never been able to do before, and that is to get our therapies out to the invasive front of the disease, realizing this is not a focal problem. And if we can somehow open the blood-brain barrier, for example, and deliver our therapies there to help them, expedite their endpoint, which is to control the disease, and I think that would be a major advance. So I would say, just in summary, that I think our goal is to try and get whatever therapies we want to use, we've got to get them out to that disease, because that residual disease is what's repopulating the tumor and causing the problems that we can't fix at recurrence.
Excellent points. And I think so often we have these, as was discussed yesterday, we have these fantastic results in preclinical models and even in early phase clinical trials, and then we stumble despite the fact that the mechanisms and the targeted therapies seem to work exceedingly well. Getting them there is part of the challenge. Dr. Brem, you've spent a tremendous amount of your research efforts and clinical efforts getting chemotherapies to the resection site. Your thoughts on the unmet needs and the next steps in terms of potential clinical trials for focused ultrasound in the context of glioblastoma?
Thank you. And I, of course, agree with Dr. Berger that is our major goal, is to get to the residual tumor in particular, and to find better ways of doing that. I think that there's really, focused ultrasound has promise in two different directions. One is to really get to parts of the brain that we want to preserve in terms of their eloquence and can't remove surgically or with direct ablation, but opening up the blood-brain barrier, making them more, accessible, and being able to do it in a way that we can monitor while we're doing it, that we're actually getting the outcome that we want in terms of better diffusion into that area. And certainly the ongoing clinical trials, or the early clinical trials now that are taking place across the country, across Canada, for opening the blood-brain barrier and at least using test substances like the platinum drugs and so on to get in, is a very, very good opening. I think that we're also interested in exploring immunotherapy, beautiful work that Mike Lim has done with checkpoint inhibitors that has not fulfilled its promise in the brain, unlike other organ systems, that maybe with better penetration and better blood-brain barrier disruption, it may work. But I also think that focused ultrasound, to me, the promise is also on the direct ablation, that there are, metastases, there are deep-seated tumors, that we're not quite ready for the clinical trials yet. I think the safety needs to be worked out a little bit better, the technology a little bit better. But ultimately, I could see that as an incredible, tool in the hands of the neuro-oncologist, neurosurgeon, to be able to directly ablate without doing a full craniotomy. So I think the potential is enormous, and we're making progress in that direction as well.
I agree with you completely with the comments, and one of the challenges I think is that oftentimes I've heard it said that you only get one real bite at the apple in terms of having the opportunity to launch a trial, and that it has to be extremely well designed, and the ability to have those preclinical data points, all the T's crossed and I's dotted before going into a large trial. Michael, Dr. Berger and Dr. Brem talked about the immunotherapy a good bit. That's your area of expertise among so many things, but your thoughts on focused ultrasound and specifically immunotherapy as a means of treatment, and what disease state frontline, recurrence, residual, might you think focused ultrasound might have the most applicability? And where do you see sort of the genesis for a future clinical trial if you were going to design one?
Thanks, Jason. So, I think, when you just take a look at any new technology, any intervention, the two questions you always ask first before you, embark on any trial is, will this help the patient in terms of survival or quality of life, right? And you don't have to have both in order to do something good for patients. And so with the technology, as Dr. Berger and Dr. Brem mentioned, you have this one avenue of potentially, improving survival, right? Which is getting at the tumors beyond the margin, tumors that you can't see, tumor cells that you can't see. And so, you have this tool, this focused ultrasound device, and as they mentioned, immunotherapy could potentially be something that works with it. And the appeal for immunotherapy is that it allows for surveillance as well as the ability for getting those distant tumor cells that you can't see. And the major barrier is that the brain, unlike a lot of other cancers, just has not been responsive. They call it the cold tumor. And there are many reasons why it's a cold tumor. People are actively looking at it based on immune subsets, heterogeneity. The brain has a different mechanisms for eliciting an immune response. And so, with focused ultrasound, a lot of people are kind of thinking about this in a concept of what we call kindling, use of some targeted ablative therapy to, cause antigen release, some sort of focal inflammation, and then you hope that you can bring in the immune cells that pick up the antigens, present the antigens, and start an immune response. And there was actually a very nice review in the pre-education session by Dr. Trimby, and I urge people to look at it because it talks about the different energy ways, the different ways to use the energy of the focused ultrasound to elicit an immune response. And so, I think it's, while it's a new technology, it's not different than other immune strategies, and that we'll probably have to be a little bit more specific. Take tumor samples, understand what's driving the immunosuppression. For example, you can look at different tumors and see it's a myeloid predominance, or you can look at other checkpoint expression molecules and then tailor the therapy to be biomarker specific. On the other hand, to improve people's quality of life, right? Inflammation, T2 changes often are the problems for patients in the recurrent setting. And that's what's causing deficits. We're putting patients on tons of steroids, making them have proximal leg weakness. And so if you could use focused ultrasound to figure out whether it's an immune response, whether it's figuring out ways to get more chemo into patients, we could do a lot of good for patients. Maybe not improve the survival, but if we can figure out ways to decrease the steroid requirements during that time, that's another avenue for clinical trials too.
Great points, and I want to emphasize it's not just the survival endpoint that we need to focus on. It's the quality of life endpoints of these patients, and it's so important to the patients and their caregivers. It takes a village. We've heard neurosurgical perspective. Manmeet, your thoughts on the opportunities, where the greatest unmet needs are, and where focused ultrasound might have the most applicability for design of a new clinical trial for glioblastoma.
Sure, thanks. So I would echo the sentiments expressed by the three previous esteemed panelists, and would say go back to the survey. And I think it's not going to be one thing, it's going to be a mixture of things. And we have to be very strategic in how we go forward, because again, as was mentioned, glioblastoma is often referred to a graveyard for drug development, right? As Ron DiPigno has said, and I think Dr. Brem said in the first session this morning. So to us, obviously, I think the need is, when we ask this question, where's the unmet need? I think it's in every single component of glioblastoma, whether it's a front setting, where Dr. Rajesh Stupp led the study and got approval for temozolomide, but we know 2/3 of our patients are unmethylated, and 9 months of temozolomide gets them 24 days worth of increase in survival. So it was significant on the study, but clinically is not as meaningful. So certainly one could look at trials in the unmethylated segment in the upfront setting to see, can we do better than what temozolomide does. We could definitely take utilization or take advantage of recurrent glioblastoma for sure, where we know that there is no single modality or treatment that has ever even shown a single day worth of increase in survival. So anything we do for our patients is pretty much palliative, unfortunately. And all the efforts we have right now are equally ineffective or effective, depending on how you look at it. So there, I think designing window of opportunity trials would be highly advisable. Drug surgery, drug studies are a nice paradigm, especially looking at some of the immunotherapies, as Dr. Lim had alluded to, especially where we don't know, right? We don't even know if we have the right target. We know in glioblastoma that unlike other tumors where high tumor mutation burden leads to utility of immunotherapies or immune checkpoint inhibitors, we have not seen that in glioblastoma, although there's a pan approval for pembrolizumab in that setting, which is tumor agnostic. What I would advise people not to do is do those smallish single-arm trials that I've been guilty of doing myself, where you don't have biomarkers or high-end imaging or some specific aspect where if you have a few patients who will do well, say 2 or 3 patients out of 20 patients who do well, we don't know where to take our trial to the next stage. And that has actually killed a lot of drug development in glioblastoma, unfortunately. Then I think as critical as what we are doing here is get these KOL panels to really think, step back, and look at what the next best clinical trial design will be. But work, and even if we are doing different things, work together in a way so that one trial may inform the others, and we can learn from what others have done and build on that.
Excellent points. And I think it is not likely going to be just one single approach or one single option. You've raised at the early onset of your commentary about unmethylated status. There's perhaps no one better to comment on the opportunities of Temodar in the unmethylated status of patients with glioblastoma than Dr. Stupp. Dr. Stupp, your thoughts on the same sorts of questions about opportunities and the next steps in terms of a trial. If you were given the opportunity to design the next focused ultrasound trial for glioblastoma, what would it look like? I think you're unmuted.
I'll take the opportunity to basically come back and change the question in a way. It's not so much how we design the trial. We find the trials and we have plenty of opportunities to do them. The unmet need in every single disease stage, we can discuss for hours, we find it. But we have to define the questions we want to answer. And from the questions, we actually come to the trial design. And we alluded to many of these things yesterday. I think we need to know when to give the drug, how long the blood-brain barrier is open, how much of the field we open. We had even discussions on the parameters. We then discussed, okay, we need to keep certain things as a fact, and we can't have 5 variables we change. And I think here what Manmeet said, we need to exchange those data. And then depending on that, I think the different trials come. But we are kind of jumping ahead. And I say, like drug company, when you have from the first idea, you have to have the marketing plan already ready. But I need to have the technology to the point that I know what to measure, what I can achieve. Of course, the unmethylated ones in the upfront setting is perfect, especially ones who were completely resected. We still have the challenge there more on the newly diagnosed is that we have to put to plan it up front before the patient even knows that he has a GBM, depending what kind of modality we use. But I think that's clearly something. But to improve, I need, and then also a target or a treatment that I really want to have to go in. And I think here we can have another 2-day session just asking which way should we go. Is it a cytotoxics? Is it immunotherapy? Is it a combination thereof? The targeted therapies? But I think we should right now focus on the technology and what we need to take this to the next level.
Excellent points, and I think there's so many unanswered questions, not just about the technology. We talked a lot about the mechanisms of action yesterday and had individual sessions on some of the more leading or more promising mechanisms of action. Dr. Chang, I'll let you have the last word. You get to the comment on the same questions, unmet needs and potential for a clinical trial. How might you conceive of one if you were to develop one for focused ultrasound and glioblastoma?
Well, thank you, Jason. And I think the prior speakers have really outlined some of the challenges and indications. I think whenever I think about a clinical trial, you always have to think about the patient selection, the treatment that you're actually going to be performing, and then the outcomes of what you're interested in looking at. And I really want to highlight what Roger said about the proof of principle is if we know that, or we think that this is how the ultrasound technology is working, how do we show that in a robust way? And I think the pre-surgical, that window of opportunity studies that are being done are really going to give us information, not only potentially in the enhancing component, but also the non-enhancing component. So I think a lot of the work that's being done to really understand the heterogeneity of the disease, not only in terms of its contrast enhancing and blood-brain barrier disruption issues, but the microenvironment, because as Michael, Dr. Lim pointed out, there's heterogeneity as well, not only in the tumor, but also in the microenvironment and the response that the normal patient, a patient would have towards having the tumor in place. And so I think the opportunity for looking at the tissue biomarkers, as well as potentially any other liquid biopsies, the imaging, really concentrating that early development study with all of the techniques that we can better understand the actual biology, not only looking at the classic PK/PD effects, but some of the other sort of novel ways to evaluate these treatments. So I think that, I think we're well on our way to doing those types of studies, but I also liked what Manmeet said about making sure that we do this as a group. So we understand where everyone is working, so there's not redundancy and that there's synergy in all the effects and the treatments that we're doing. So we can understand, putting it all together, what's the true value and impact that this technology might have for our patients. Thank you.
Well, excellent points by all speakers. I think I've gained some answers, but perhaps just as many new questions now as I've heard all of you talk. So the opportunities to join the next session, I think, will be occurring in just a couple of minutes. I thank all of the discussions for providing their insights, and hopefully we shed a little bit of light on the burning questions around this symposium. Thank you so much.